Hi everybody. So yesterday we talked about regular genetic engineering, where we looked at recombinant DNA technology and how you can manipulate a DNA molecule to contain a gene. And usually that's moving a gene from one organism to another, like moving jellyfish DNA into rats or bunny rabbits. Today we're going to take that a step further because a recent development in the field of genetic engineering is called extreme genetic engineering. It's also known by the name synthetic biology, which is probably a more appropriate term for the science because it means that you are generating code for creating new proteins that didn't exist before. So you're synthesizing or creating new code that never existed before. Some people abbreviate that synthetic biology to SynBio. So let me first explain kind of how that works. What you need to understand is that DNA, while it's an important molecule, it's a storage molecule. It doesn't actually contain, or it isn't actually not involved in the production of proteins in the cell. It's the code for proteins, and proteins do everything in your cells. But it's not really used in making those proteins. Instead, you need a new molecule, RNA. Okay, so the code from DNA gets rewritten onto an RNA molecule. And so if you understand the genetic code and how it works, you can actually manipulate that genetic code, not by moving a code from one organism to another, but by actually creating your own brand new code. So to understand that, let's look at the RNA molecule first of all. RNA, unlike DNA, is single-stranded, so you only have one side to the molecule, and it also has a backbone that I've just highlighted there for you. That backbone is made of a repeating phosphate and sugar segment, and if none of this is really clicking too well, don't worry about it right now. You're going to get into it later on. But understand that what's important here is the code is written in the sequence of A's, U's, G's, and C's that you see in our line right here. This is the code right here. In other words, if there's an adenine on the molecule, a specific molecule called adenine, the code reads A there. If there's a uracil on the molecule, the code will read U. So DNA's information is rewritten into RNA, and then RNA takes that information and uses it to construct the protein at the ribosome in the cells. Okay, that's how it works. And you can see here that each code in the RNA molecule codes for a specific amino acid. If you've had biology, you've probably worked with these codes before. At any rate, it's a really simple code, and that's the point. The code is so simple, and it's only 4 letters, 4 possibilities. You can arrange those letters in any way you want to create a brand new code, and that's the basis of synthetic biology. So let's look at some of the implications of synthetic biology so far. Okay, first of all, synthetic biology has been used. The first synthetic biological organism was created in 2010, and it was created by inserting a computer-generated code that they then synthesized in a lab into a bacterium. Since then, Goodyear and Michelin have been working to take bacteria and use that bacteria to write a code to get the bacteria to produce isoprene. And isoprene is a component of tires. So they're working on that. Another company, ExxonMobil, is working to take algae and use that algae, try to insert a code to get the algae to start producing biofuel. Okay? So there are a couple companies that are actually actively working on this. Here's the thing. In 2012, the Obama administration set up a special committee, and that committee looked at the field of SynBio and decided to let the industry regulate itself, which prompted a call by lots of organizations for a moratorium, in other words, a stop to letting the industry regulate itself and a stop to all research in the field until scientists can debate some of the ethics of the issue. The biggest voice against SynBio is an organization called Friends of the Earth, and what they believe is that we need to slow down on this technology. They feel like the technology and our capability to do all of these things is outpacing the research that we need to understand what kind of risks this might pose to the environment. They cite the fact that when genetic engineering first began, they had lots of unforeseen problems with that technology as well. And now we've been doing genetic engineering for decades, and it works pretty well. But this is new, synthetic biology is new, and they argue that we need to slow it down a little bit and that there could be some repercussions to the planet that we just haven't thought about yet. So that's kind of how you do it and why you do it, and some of the issues against, or some of the voices against synthetic biology.